On behalf of the investigators, it's a pleasure to present to you the results of a retrospective study of treatment with substrate enhancement therapy with deoxycytidine and thymidine conducted in patients with thymidine kinase 2 deficiency. On the next slide are the disclosures for the authors. This study was funded by Modus Therapeutics, a wholly owned subsidiary of Zogenix. Next slide. Thymidine kinase 2 or TK2 deficiency is an autosomal recessive disease. It is a disease of mitochondria, and the gene is encoded in nuclear DNA. TK2 is caused by a defect in the TK2 enzyme, which is critical for the first step in phosphorylation of the pyrimidine nucleosides DC and DT. Ultimately, the triphosphates are needed for incorporation into mitochondrial DNA. So a defect in the initial phosphorylation step results in a reduction in mitochondrial DNA, a reduction in components encoded in mitochondrial DNA, such as the electron transport chain, which is needed for energy metabolism within cells. The disease is ultra rare, and as you will see shortly, only about 100 patients have ever been reported in the literature with TK2 deficiency. Most patients present at a young age with rapidly progressive disease, leading quickly to hypotonia or tetraparesis, respiratory failure requiring ventilatory support, and death. Less commonly, patients may present in adolescence or in adulthood with a slower disease course. In these cases, there's progressive muscle weakness and often the need for non-invasive ventilatory support, such as BiPAP at night. In animals, treatment with pyrimidine nucleosides restores mitochondrial function by working through salvage enzymes and/or residual amounts of TK2 enzyme. On the next slide, you see data from the 38 TK2 patients in the retrospective study that was gathered prior to treatment. This display shows the number of patients losing a particular milestone in each column. So a single patient may be represented across multiple columns if there was more than one milestone that was lost. The milestones lost are displayed from left to right, with the more advanced ones to the right. So you see the ability to hold the head upright at the left and running toward the right. We also included the ability of the patient to dress and bathe. Clearly, untreated TK2 deficiency is characterized by significant loss of motor milestones. Our investigators tell us that in the absence of treatment, the disease is always progressive. The next slide shows study design and demographics. In the retrospective chart review study, MT1621-101, we gathered data on 38 pediatric and adult patients with genetically confirmed TK2 from 8 clinical sites in 3 countries. All patients were treated with chemical-grade nucleosides or nucleotides for a median of 1.5 years, ranging from 92 days to approximately 7 years. Patients were treated under compassionate use or in an investigator IND. You can see on the right that we also gathered information on untreated patients by doing a comprehensive literature review. We identified 103 unique patients with TK2 deficiency who were not treated and gathered survival status and functional outcomes. As age of onset is a key factor in prognosis, we categorize patients by age of onset. Recall that patients with the youngest age of onset progress rapidly and often die, which you can see from the large proportion of deaths in the untreated patient group. In contrast, none of the 38 patients who were treated have died. These next several slides show results. We looked at data in several ways, going from a population level analysis, looking at survival, to looking at multiple assessments across patients in what we call a domain analysis, to looking at milestones, functional status, and functional assessments in individual patients. This is a summary of the analytic approaches we took for survival. We compared the survival of the 38 treated patients with an external control data set taken from a subset of the untreated patient group, so that it included only those who had a known date of death or age last known alive, and were alive at the age at which the youngest patient was treated in the treated data set. We did 2 basic types of analyses, a whole population analysis, which includes all patients, and a matched pair analysis, which matches a single treated patient to a single untreated patient according to a conservative algorithm. You can see that both of these analytical approaches are highly significant with small p-values, suggesting that treatment improves survival. On the next slide shows that we use a domain approach to integrate results from many tests that a single patient may have had. We base the threshold for improvement on minimal clinically important differences from other neuromuscular diseases, as well as gain of motor milestones or a change in status. For respiratory, the threshold for improvement included things such as a decrease in ventilatory support of at least 4 hours or coming off a ventilator. And for feeding, it included coming off a feeding tube. Most patients showed improvement in the motor domain and showed either improvement or stability across all domains. We took the sum of all domains for the overall. For a disease that progresses without treatment, seeing the high percentage of patients who are stable or improved is a big win. The next slide shows individual patient data on motor milestones. Recall the earlier slide I showed of milestone losses. This is regains or new gains in milestones. We call regains those milestones that the patient initially acquired and then lost prior to treatment and then acquired them after treatment. 17 patients regained milestones. This supports that treatment reverses the progressive disease course. New gains are milestones that the patient did not have before treatment and were gained after treatment. 6 patients had new gains in milestones. No patients lost milestones after treatment. We also had supportive findings and endpoints that were measured, such as the 6-minute walk. In terms of respiratory status, one patient who had a tracheostomy and was ventilated was able to come off the ventilator completely, and we had 3 patients decrease the number of hours of ventilation required by at least 4 hours a day. 3 patients had improved dysphagia and oral intake and had their feeding tubes removed after treatment. Next slide. The drug is given orally as a solution. The most common adverse event was dose-related diarrhea. The diarrhea resolves over time with dose reduction and continued treatment. Most patients are able to reach the target dose of 400 milligram per kilogram per day. There were 2 patients who discontinued treatment due to elevated transaminases. These patients did not have elevated bilirubin and therefore did not satisfy the definition for drug-induced liver disease. The elevated transaminases resolved after discontinuing the drug. We also had about 1/3 of the patients who had elevated ALT that decreased after treatment. Hepatic involvement has been seen with TK2 deficiency, and the decrease in ALT after treatment is consistent with the treatment effect. In summary, TK2 deficiency is a severe progressive disease which causes significant morbidity and mortality. A retrospective chart review of 38 patients treated with pyrimidine nucleosides showed improved survival and also improvements in motor, respiratory, and feeding function. The majority of patients were either stable or improved after treatment, and this was seen across all ages of onset. The oral treatment was generally safe and well tolerated, and dose-related diarrhea was the most common adverse event. Finally, Modus Therapeutics is developing this approach, substrate enhancement therapy, as a treatment for patients with TK2 deficiency. I would like to acknowledge all of the investigators and collaborators for the MT1621-101 study. Thank you.
Hello, everybody. My name is Tolga. I'm a Senior Medical Director in Astellas Global Development Team. And today I would like to present you ASP0367 program and provide updates on our ongoing clinical trial activities for primary mitochondrial myopathies. First, I would like to thank organizers and UMDF for giving us the opportunity to present our development program in their first virtual meeting. Thank you. Here are our disclosures and disclaimer. Astellas is a global biopharma company with Japanese roots with more than 100 years of drug development history. Astellas operates globally, develops and markets a number of compounds in various therapeutical areas, including oncology, urology, transplant, and infectious diseases. Following our new mission, becoming the leader in developing therapeutics that treat a wide range of serious diseases by modulating mitochondrial function, Astellas co-founded and later acquired a biotech company, MitoBridge, and further identified mitochondrial research and development as one of the 5 primary focus areas. This move enables us to support development of diverse pipeline and preclinical programs and advancement of multiple programs into global clinical development in mitochondrial research field by leveraging global medical, regulatory, and marketing capabilities of a big pharma. Peroxisome proliferator activated receptors, PPARs, are nuclear receptors that act as transcription factors controlling wide range of biological processes by regulating gene expression. PPAR delta is highly expressed in various tissues in the body, including skeletal and cardiac muscle. It has been shown that PPAR delta regulates the synthesis of new mitochondria, energy generation by fatty acid oxidation, and improves muscle endurance. Astellas is currently investigating a new class of PPAR delta modulators, which are the product of many years of preclinical research and collaboration with a MitoBridge co-founder, Ron Evans from Salk Institute. ASP067 is an oral, once-a-day, highly selective PPAR delta modulator, which is currently being investigated for the treatment of fatigue and exercise intolerance in patients with mitochondrial myopathies. ASP067 mediated gene induction is being investigated to lead to an increase in mitochondrial fatty acid oxidation, as it is shown in upper right-hand figure in red, in cells obtained from patients with mitomutations with defective mitochondrial fatty acid oxidation. In addition, further, in DIO mouse model, 4 weeks treatment of ASP0367 decreased running fatigue, as it is shown in lower right-hand figure in red, along with increased fatty acid oxidation genes in skeletal muscle in this model. In the clinic, safety, tolerability, pharmacokinetics, and pharmacodynamics of ASP067 were evaluated in about 100 healthy volunteers in single and multiple ascending dose studies in phase 1. Safety and tolerability profiles of ASP0367 were similar, comparable to placebo. Pharmacokinetics of ASP0367 achieved target range at doses of 10 milligram a day and above. Further, PPAR delta target gene expression biomarkers, which is shown here with an example of one of these genes, C1A gene expression, were induced at the doses above 10 milligram a day, indicating biological activity and further target engagement of ASP0367 in human. This year, Astellas is launching 2 simultaneous clinical studies with ASP0367, targeting 2 myopathies in which mitochondrial functional deficits are either the primary or secondary factor in disease pathology. The first one is our upcoming phase 1b study for Duchenne muscular dystrophy, and second is our planned seamless phase 2-3 study for primary mitochondrial myopathy. Astellas has the second PPAR delta modulator, ASP1128, which is currently being investigated for treatment of acute kidney injury, which is currently in phase 2 of clinical development. In coordination with FDA and working closely with key opinion leaders in the field, Astellas is planning a safety, tolerability, pharmacokinetics, and pharmacodynamics study in primary mitochondrial myopathy patients, leading to a randomized, double-blind, placebo-controlled adaptive phase 3 study with open-label extension period. A total of 190 adult and pediatric participants in separate cohorts will be enrolled in approximately 15 sites across the United States. Participants will receive ASP0367 and placebo once a day orally for 52 weeks during the double-blind period. Double-blind treatment period will be followed by an open-label treatment extension in which all subjects will be offered ASP0367 for 24 weeks. Study is planned to start first quarter of 2021. In order to be eligible for our study, participants' primary mitochondrial myopathy diagnosis will be confirmed by the presence of molecular genetic abnormality, nuclear or mitochondrial, causing mitochondrial dysfunction. Associated symptoms such as muscle weakness, fatigue and exercise intolerance, and myopathic physical examination findings. Participants will be required to follow the study requirements for the length of the study, including performing motor function tests such as 6-minute walk test, as well as the use of digital applications and video recordings. Efficacy of ASP0367 on endurance and fatigue will be evaluated by distance walked in 6-minute walk test and patient-reported fatigue scales, respectively, by the end of 52-week double-blind portion of the study. Exploratory remote primary mitochondrial myopathy video assessment will be recorded throughout the study, along with qualitative audio interviews in order to collect direct feedback about participants' experience in the study, particularly with respect to treatment and changes in disease burden. Further, muscle and cardiac imaging will be assessed before and after the treatment in a sub-study at the specific clinical centers. Thank you very much for your time and attention. We look forward to working closely with this community during the progress of our development program. Thank you.
Good afternoon. First, I'd like to thank the organizers for the opportunity to present today. My name is Chad Glasser, and I'm a director of clinical research at Cyclerion Therapeutics, which is a small biotech company located in Cambridge, Massachusetts. Cyclerion has an expertise in the nitric oxide soluble guanylate cyclase cyclic GMP pathway. We've developed a portfolio of tissue-targeted molecules that stimulate this pathway. The program that I'll be speaking to today is our central nervous system, or CNS, targeted stimulator, IW6463, and our plans to conduct a phase 2A study in individuals with MELAS. So what is our rationale for evaluating a CNS-penetrant soluble guanylate cyclase, or SGC, stimulator in this population? Well, we know that signaling through this pathway, which is shown here on the left, plays an important role in mitochondrial biogenesis and function, as well as neuroprotection and vascular homeostasis. These are all potential downstream therapeutic effects associated with the generation of cyclic GMP through this pathway. Although we know that mitochondrial disease encompasses a wide range of mutations and clinical phenotypes, the majority of individuals with mito, approximately 80%, experience CNS symptoms. It's also well documented that many of these individuals have a deficiency in nitric oxide, otherwise known as NO, and therefore impaired NO signaling. This point is further supported by the Mitochondrial Medicine Society's recommendation that precursors of NO, like L-arginine, are used in MELAS. As you can see on the left, 6463 intervenes at a different point in the pathway compared to arginine. By binding to the central enzyme SGC and driving cyclic GMP concentrations at the source, unlike arginine, 6463 is able to amplify endogenous NO signaling with no attenuation of response. Before I introduce our clinical program, I wanted to briefly share some of our preclinical data. This slide summarizes the positive effects that 6463 has demonstrated in preclinical studies across what we're calling 4 key domains of neurodegenerative disease. Compared to peripherally restricted stimulators, 6463 improves cerebral blood flow in areas associated with memory and arousal, as measured by fMRI bold imaging. Specifically important to the underlying pathophysiology of mito, 6463 increased ATP and restored expression of already depressed genes and cells from individuals with mito. In vitro studies, 6463 decreased markers of LPS-induced neural inflammation. And finally, related to neuronal function, it restored hippocampal long-term potentiation to wild-type levels in a neurodegenerative mouse model. It prevented the decline in hippocampal spine density in aged animals and also improved cognitive performance in rodent models with cognitive deficits. Because of this multi-dimensional pharmacology and the potential downstream therapeutic effects of increased levels of cyclic GMP, we believe that 6463 may have the potential to positively impact diseases with deficient NO signaling, such as mitochondrial disease, by multiple mechanisms. By restoring the signaling of this key central neurotransmitter system, we have the potential to address impaired blood flow, mitochondrial dysfunction, neural inflammation, and neurodegeneration, thus potentially improving overall brain health. Taken together, these preclinical findings supported the initiation of our first-in-human study in healthy volunteers, which we completed earlier this year. So this was a 3-part randomized placebo-controlled study conducted in 110 participants. The main objectives were to evaluate the safety, tolerability, and pharmacokinetics of 6463, but we also wanted to assess markers of target engagement and evaluate 6463's ability to cross the blood-brain barrier. Single and multiple ascending doses over the course of 14 days were administered to participants across a wide dose range, and the pharmacokinetics were also evaluated with and without food. Overall, 6463 was very well tolerated at all single and multi-dose levels. There were no discontinuations due to adverse events, no SAEs, and all adverse events reported were mild in severity and resolved before the end of the study. Its PK was linear, predictable, and consistent with daily dosing, and it was determined that it can be taken with or without food. We also obtained CSF samples from participants to confirm the presence of 6463 and observed mild changes in blood pressure consistent with our mechanism, confirming target engagement. The results from this study allowed us to select a clinical dose that is safe and well tolerated and expected to achieve steady-state CNS exposures in the targeted therapeutic range. This target was based on the exposures associated with the positive preclinical CNS pharmacology, which I shared previously. For our first study, although this molecule may have potential to impact CNS manifestations across a wide range of mitochondrial diseases, we have decided to first focus on individuals with MELAS. I'm sure I don't need to explain in detail what MELAS is to this audience, but it's a genetically and phenotypically defined mitochondrial disease characterized by NO deficiency and with no currently approved treatments. Although disease presentation commonly varies, the cardinal symptoms of MELAS involve the CNS. This includes seizures, dementia, headaches, learning disabilities, altered consciousness, and of course, stroke-like episodes, among others. One of the key reasons we chose individuals with MELAS for our first study in Mito is because of the clinical precedents that exist regarding this pathway. As I mentioned previously, intravenous arginine, an NO precursor, is recommended as an acute treatment for stroke-like episodes. Also, the use of oral arginine or citrulline to prevent these episodes has anecdotally been shown to provide benefit, though no definitive studies have been performed. Even with anecdotal reports of efficacy, these treatments still come with their own limitations, including poor pharmacokinetics and issues with tolerability and compliance. As for pathophysiology, individuals with MELAS often have impaired blood flow in the brain, inflammation, angiopathy, and endothelial dysfunction. Much of this pathophysiology has also been linked to the cardinal CNS symptoms of this diagnosis. The preclinical pharmacology, which I discussed previously, aligns very well with these findings, particularly 6463's impact on mitochondrial function and cerebral blood flow. So we're actively working to initiate a phase 2A multicenter open-label pilot study in individuals with MELAS. Our primary objective is, of course, safety and tolerability, but we're also very interested in measuring 6463's pharmacodynamic effects and impact on biomarkers associated with 4 key clinical and pathophysiological aspects of MELAS shown here on the right. To evaluate our impact on mitochondrial dysfunction, we are assessing a range of biomarkers, including GDF-15, and most importantly, lactate, which we will be measuring both in the plasma and in the brain directly using MRS. We plan to implement innovative imaging techniques to measure any impacts on the dysregulated brain perfusion observed in these individuals. We also plan to evaluate biomarkers of neurodegeneration, including NFL, and measure any potential changes in cognitive function and fatigue. One of the challenges that we were well aware of when designing this trial is that this population is heterogeneous. It was therefore important that we utilize an enrichment strategy that could potentially improve our ability to detect an impact on disease, but wasn't too defined to the point that we couldn't enroll the trial. We settled upon individuals with neurological features of MELAS, which ensures that they have the phenotype and not just the genetic mutation, as well as elevated lactate, which attempts to select for individuals with active disease. An elevation in lactate at baseline also increases the probability that we can measure an improvement in this and other biomarkers of mitochondrial dysfunction over the course of treatment. Participants will receive up to 29 daily doses of 6463 and will complete a total of 6 visits 2 of which actually can be completed at home. Because of where 6463 intervenes in the pathway, it has the potential to work synergistically with endogenous INO signaling and INO precursors. Participants may therefore continue taking concomitant arginine or citrulline while being enrolled in this trial. We're targeting to enroll approximately 20 participants and are excited to be working with many of the top mitochondrial disease centers in the US. But just wanted to say thank you to the organizers once again for the opportunity to share our program today. We look forward to further collaborating with the mito community, including researchers, clinicians, and of course, those living with mito.
Hi, my name is Alex Dorenbaum. I'm the Chief Medical Officer at Rainier Pharmaceutical, and I want to thank the organizers of UMDF for inviting us to talk to you. Reneo is a clinically stage company focusing on genetic mitochondrial diseases. We began operations in 2018 with a very experienced management team. Our drug, Reneo-1, is a potent selective PPAR delta agonist that is being tested for treatment of patients with primary mitochondrial myopathy. And the way Reneo works is we went to look for the potential drugs that could be used in patients with PMM, and we looked at all the companies that had worked with multiple drugs and that they were currently not developing those drugs because of business reasons. And we look at the biology of those drugs and try to understand if that drug may work in patients with PMM. And the advantage of doing this is that Big Pharma usually does a lot of studies with these drugs before they actually give them to patients. And Reneo-1, when we evaluated Reneo-1, it already had an extensive safety pharmacology and a chronic toxicology study that had been completed in animals. So we had a lot of understanding about how this drug may be tolerated and how it may work in patients with PMM. In addition to this, the drug already had quite a bit of information in regards to the chemistry, the manufacturing, and the quality control of the drug. And this is very important because when you do studies, you want to make sure you have a consistent, good medication that you can use in your studies. We also knew that the drug had a favorable pharmacokinetics or PK profile. This is very important because PK or pharmacokinetics refers to how a drug is absorbed into the body, metabolized or used in the body, and then excreted out of the body. And the advantage of this drug is that it can be given once a day by mouth, very easy to take, very simple procedure to be able to take the medication. And also, the drug already had extensive phase 1 and proof of concept information, and that enabled us to do a PMM clinical study very quickly. And we actually have completed that proof of concept study in PMM. And let me share a little bit about that program with you. So one of the main reasons why we got interested, in PMM and in Reneo-1 as a drug that can help PMM is because Reneo-1 is what we call an agonist. It stimulates a receptor in the cells. And the receptor that Reneo-1 stimulates is called PPAR delta. And when you stimulate PPAR delta in the cell, and by the way, PPAR delta is primarily found in muscle cells. So when you stimulate PPAR delta, what you do is you induce the formation of new enzymes in the muscle that help remodel the muscle so that it will work better. What it does is it actually increases the enzymes that are available in the mitochondria, the enzymes that metabolize fats to generate energy. And it also stimulates the genes that are involved in the formation of new mitochondria. So this was all very exciting for us because if we can improve how the mitochondria function and we can expand the number of mitochondria in the cell, we can provide better energy for the muscle to work well. And that's the suggested mechanism of action of PPAR delta agonist drugs. So this made a lot of sense to us, and that's why we were very excited about this drug. And then we set up to do a first study, and the first study that we did was a proof of concept trial in patients with PMM. This was a 12-week study. Patients received the medicine once a day by mouth, and they had the option to continue into an extension study after the 12 weeks of treatment. We enrolled about 23 patients in the study. All of these patients had mitochondrial gene defects. They all had a history of myopathy, that is injury and damage to the muscle and difficulty in moving their muscles. They all were adults, older than 18 years of age. And because we wanted to evaluate exercise tests in these patients, we made sure that all of these patients could have the ability to complete a walk test. And the type of studies that we did in this clinical trial were in two main groups. First, we did exercise tests, and second, we did symptom questionnaires to look at how patients feel as well. In the exercise tests, you can see there we did at least 3 tests: the walk test, the peak exercise test, the submaximal exercise test. And the idea here was that we don't plan to use all of these tests in future trials, but we wanted to see how each of these tests behaves in the patients and whether patients can complete them and what kind of information will these tests give us. And based on that, we decided to choose the walk test as the test that we plan to use in future clinical studies. In addition to that, the symptom questionnaires that we use are common questionnaires that are included in clinical trials, like the SF-36, that is a quality of life questionnaire. We also used the modified fatigue impact scale that primarily evaluates fatigue in patients with chronic illness. And we looked at the brief pain inventory test that looks at pain in chronic conditions as well. And we used all of those tests because when we interviewed patients with PMM, they told us that the main symptoms that they have are lack of endurance, difficulty doing activities that require physical exercise, etc., etc. And here is a summary of the results of that proof of concept trial. The drug, PMM, was safe and well tolerated by the patients with PMM. Of course, I need to caveat this by saying this is a small study, but we anticipate the drug is likely to be tolerated well because it has been used in multiple studies, and in all of those studies, the drug was well tolerated as well. There were promising improvements in the walk test and in the symptoms questionnaires, and the majority of patients who took part in the study chose to continue treatment in the extension part, and that was very reassuring to us. In addition to this, the data that we obtained from the study was sufficient to inform the design of the next, what we call pivotal trial, which will be the main study that we would use to talk to regulatory agencies to get the drug approved for use in patients with PMM, should the drug prove to be efficacious. And here is, in broad terms, the design of the study that we are currently planning. This will be a 24-week, double-blind, placebo-controlled trial. Double-blind means the doctors don't know what drug they're giving to the patients, and the patients also don't know if they're receiving the drug or a placebo, or perhaps 1 or 2 different doses of the drug. In this study, actually, we are planning to use 2 different doses of Reneo 1 and a placebo. So what that means is that out of every 3 patients, 2 patients will receive medication and 1 patient will receive a placebo, which is a dummy medication. The study will include about 200 patients with mitochondrial gene defects. This will be an adult study in patients older than 18 years of age and with a history of myopathy. Again, because we're doing exercise tests, we want patients to be able to complete the walk test. And we plan to include about 20 centers in North America, in Europe, and in Australia. Based on the results of the previous study that we did, we plan to use a walk test and also the symptom questionnaires as the measures of efficacy or outcome in the upcoming trial. And this is my last slide. Currently, as you know, a lot of the research is being halted because of the COVID epidemic, but that hasn't stopped us from doing our work, even though we cannot quickly bring patients to the clinic to do the research right now. What we can do is to work with the regulatory agencies in the different countries where we plan to work and start doing all the process to get the approvals to be able to do the studies in those countries. We're also working to select the clinics that will participate in the trial and to make sure that we do all the paperwork and all the necessary agreements that need to be made so that a center can participate in the trial. As you know, each center has to go through what is called the ethics committees or the institutional review boards or IRBs that review the studies to make sure that it is okay to do the studies in the patients. And last but not least, we are hoping to be able to start the study around the first months of 2021. That's only 6 months from now. It's around the block, so we're working very hard to set up everything to be able to start the study very quickly as soon as the COVID epidemic permits. We anticipate the study will start very early in 2021. Well, it's been for me a pleasure to be able to share with you the Reneo programs. And I want to thank again the UMDF for giving us the opportunity to be part of this session. And I look forward to hopefully meeting you all in person in the near future. Thank you again.
I thank the organizers for allowing me to speak today as the lead investigator of the upcoming Entrada MNGIE Natural History Study, ET-NHS 901. MNGIE, or mitochondrial neurogastrointestinal encephalomyopathy, is a lethal autosomal recessive disease characterized by mitochondrial abnormalities, including multiple deletions and depletion of mitochondrial DNA, leading to ptosis, ophthalmoplegia, severe gastrointestinal dysmotility, cachexia, peripheral neuropathy, and leukoencephalopathy. In the classic form of the disease, onset is in the late teens, with gradual progression over 2 decades, leading to fatality at a mean age of 35. The disease is caused by TYMP mutations that cause severe deficiency of thymidine phosphorylase, or TP. TP resides in the cytoplasm, where it normally degrades the nucleoside thymidine into the base thymine and the sugar 2-deoxyribose 1-phosphate. In patients with MNGIE, there are dramatic elevations of thymidine and deoxyuridine in plasma, as shown here, as well as tissues. In the classic form of MNGIE, levels of thymidine in plasma average 8.6 micromolar. In contrast, healthy controls and asymptomatic TYMP mutation carriers have no detectable thymidine or deoxyuridine in plasma. In addition to the classic form of MNGIE, there's a late onset form with onset in the 40s, with slower progression and prolonged survivals into the 50s and 60s. These patients have milder deficiency of TP and less severe elevation of thymidine with a mean level in plasma of 1.3. Thus, if one could lower the level from the 8.6 micromolar concentration found in the classic form of MNGIE to plasma levels of less than 1, one could convert a patient from the classic form to a milder, slower progressive form of the disease with prolonged survival. In fact, this concept has been demonstrated through bone marrow and liver transplantations, both of which have successfully lowered levels of thymidine and deoxyuridine in plasma and led to clinical improvements. Unfortunately, both of these transplant approaches have limitations, including lack of suitable donors and severe side effects of the immunosuppressive therapies. As an alternative, Entrada is developing a proprietary endosomal escape vehicle technology to deliver thymidine phosphorylase to the cell membranes where they are engulfed by endosomes, which then bud and release the EEV products into the cytoplasm, as demonstrated here in this these immunofluorescent images. You can see the EEV product labeled in red that co-localizes with endosomes. The EEV migrates to the buds where they're subsequently released into the cytoplasm. We've demonstrated efficacy of the ENTR-501 compound, which contains thymidine phosphorylase, using our mouse model of MNGIE, which has markedly elevated levels of thymidine in plasma in the micromolar range, similar to the classic MNGIE patients. And we can see here that after a single injection of ENTR-501, you can see dramatic decreases of the thymidine level shown in purple to levels that are below the normal levels in mice, wild-type mice, and approaching the levels in normal humans. And this reduction in thymidine can be seen after intravenous or subcutaneous injections and lasts more than 48 hours. In addition, after single injections of ENTR-501 in rats, there was sustained depletion of thymidine for a week. As an initial step in the development of this compound, Entrada is sponsoring a natural history study, which is a multi-center international study, which includes Columbia, Baylor, Colorado, and other sites to be determined. Patients will be followed every 6 months for up to 6 years with a primary objective to evaluate the clinical course of the disease, with secondary objectives to look at biomarkers and additional clinical endpoints and functional outcome scales. With that, I'll end, and thank you for your attention.
Hi, I'm Magnus Hansson. I'm Chief Medical Officer of Abliva. And I will talk today about our projects and goals to deliver mitochondrial health for mitochondrial disease patients. Abliva is a small pharma company. We're based in Sweden, in Northern Europe, but we have activities around the globe. Our mission at Abliva is to develop new innovative medicines for patients with primary mitochondrial disease to improve their lives. We have one project, which I will focus most on today, KL1333, which is in clinical development stage, so in human studies, and then a couple of more projects in earlier development stage. And each of these projects focus on different aspects of mitochondrial disease pathology, and each of them have unique mechanisms of actions. So going a bit deeper into the background for KL1333, as you know, mitochondrial diseases are caused by a multitude of genetic mutations, which cause a dysfunction of mitochondria. And in particular, and quite common, a dysfunction of the electron transport chain. And one consequence of this dysfunction in the electron transport chain is a reduced conversion of a molecule called NADH to NAD+. And that leads to a disbalance, so an abnormally high NADH and an abnormally low NAD+. So why is this important? Well, NAD+ is a key molecule in regulating the energy metabolism in the cell, both short-term, but also long-term. So a potential solution to this problem, which we are exploring with our compound KL1333, is to rebalance this ratio of NADH to NAD+. So reducing NADH and elevating NAD+. And the way we do that is by KL1333 Just like NADH and NAD+, it can shift back and forth between a reduced and oxidized state, and it uses some of the NADH in the cell in this process. So the goal with this project is to restore the energy balance in the cell. And this is also important long-term, because NAD+ is a key component in many processes leading to what is called mitochondrial biogenesis. So mitochondrial biogenesis is the buildup of new mitochondria. So the aim with this project is to restore the energy balance in the cell and thereby counteract the debilitating manifestations of mitochondrial disease, such as fatigue, muscle weakness, and metabolic dysfunction. So where are we in this project? Well, we have just completed a couple of phase 1 studies. So phase 1 is the first phase of human studies, where we evaluate how multiple doses of our compound, how well they are tolerated, and how often they need to be taken to stay at an adequate level in the blood. So we just completed these studies in healthy volunteers, and we're just about to start an additional part of the phase 1 package in primary mitochondrial disease patients when COVID-19 hit us all. But we hope to continue this part of the phase 1 studies after the summer. The next step, which we're very excited about, is a randomized placebo-controlled efficacy study, where we really will evaluate how well this concept works. So this is a study which is planned for next year. So we hope to be able to start the study in the first half of 2021. We will focus on patients with MELAS mid-spectrum disease, or CPEO KSS spectrum disease, with multiple involvements throughout the body. So we are very excited about this development, and we will be able to provide more specific details later this year. So with that said, I would like to thank you all for listening. Thank you.
Thank you very much. I'm excited to share with everyone an update on our 743 refractory mitochondrial epilepsy trial, which we'll be initiating in the next few months. First, a quick update. For those of you who aren't aware, EPI 743 is now PTC 743. As many of you may know, BioElectron's assets were acquired last year by PTC Therapeutics, and the BioElectron team and all of BioElectron's drug candidates, including EPI 743, are now part of the PTC family. PTC is a rare disease-focused global commercial biotechnology company with approved drugs that are being sold around the world. PTC brings added muscle and experience to the development of EPI 743 and other compounds from the BioElectron pipeline for patients with mitochondrial disease. So we're very excited now to be part of the PTC family, and we believe now that we have an even greater chance of success in developing PTC 743. Just a quick overview on PTC. It's a multi-platform, diversified company with an explicit focus on rare disease. We have a number of different platforms developing drugs for a number of different rare diseases for both children and adults. As you can see, the BioE platform includes all of the drug compounds from BioElectron and our two lead programs, PTC 743 for epilepsy and PTC 743 for Friedreich ataxia, two potential registrational trials that we'll be initiating this year. Just for background on PTC 743, its target is the enzyme 15-lipoxygenase. Its mechanism of action is to regulate the process of inflammation, oxidative stress, glutathione depletion, and cell death that is a fundamental component of all mitochondrial disease subtypes. It's an orally administered drug. It's been in development for a number of years and has garnered a number of orphan and fast-track designations. And right now, its two lead indications are refractory mitochondrial epilepsy and Friedreich ataxia. To date, over 500 patients have been treated with EPI 743, with the longest exposure being over 10 years in a child with Lee syndrome secondary to SERP1 mutation. So just a brief overview of refractory mitochondrial epilepsy and the rationale for the 743 mitochondrial epilepsy program. As many of you know, About half of all patients with mitochondrial disease have seizures, and most of those seizures are not well controlled by traditional anti-epileptic medications for a number of reasons, including the fact that traditional anti-epileptic medications do not target the energy pathways that lead to seizures in mitochondrial disease. Furthermore, it's also well known that a number of anti-epileptic medications actually increase oxidative stress, which can exacerbate the overall pathology of mitochondrial disease. There's a strong scientific and clinical rationale for our advancing 743 for this indication. We've done a number of studies in our laboratories, and the literature is also filled with reports linking the target 15-lipoxygenase and its target pathway with refractory epilepsy syndromes. In terms of clinical rationale, we've observed in a number of our clinical studies over the years that 743 has had an important impact on refractory status epilepticus, seizure frequency, as well as seizure-related morbidity. On this slide, we demonstrate some of our in vitro data demonstrating that PTC743 can rescue cells from mitochondrial disease patients with seizures from an oxidative stress insult. In each of these 3 different patients' cells, we were able to show that 743 potently prevents cell death when cells are subject to an oxidative stress insult. This is a very reliable laboratory-based test system for us to demonstrate a potential benefit of PTC743 in the clinic. An important impact on seizures, including no seizures in patients traditionally who had seizures as part of their mitochondrial disease, and reports of seizures being under better control, and again, another report of no clinical seizures in 6 months after starting drug, and the fact that patients with POLG or disease with seizures are stable and improving on 743, which is clearly a departure from the natural history of disease. We've also collected data from our study in this mitochondrial epilepsy subtype of PCH6, which is an ultra-rare mitochondrial disorder, which results in seizures that start in the first days to weeks of life and death in early childhood. What we observed in this study was a decrease in seizure frequency, the ability to affect or disrupt the occurrence of refractory status epilepticus and prevent its recurrence. And as you see on the right side of the slide, we've been able to demonstrate a significant decrease in disease-related hospitalization in treated patients, as well as a reduction in risk of mortality in 743 treated patients. So we now stand ready to initiate our mitochondrial epilepsy phase 2/3 trial. The protocol has been approved by regulatory authorities. It's going to be a randomized placebo-controlled trial in which we're going to enroll 60 patients with study sites already identified in the United States and Europe. It'll be a 7-month study, with the first month being dedicated to ensuring that participants have a certain threshold number of seizure events, and then it'll be a 6-month period of time during which patients will receive either 743 or placebo for 6 months. And after that period, all patients will then receive 743 for the following year. The primary endpoint will be reduction in observed motor seizure frequencies, and we're going to have a special electronic diary that families are going to use to enter on a daily basis the number of seizures. And we're also going to collect important secondary endpoints, including occurrence of status epilepticus, number of disease-related hospitalizations, rescue medication use, as well as caregiver burden. We're planning to initiate this study in Q3, that's sometime between July and September. And really the start of the study is going to be dictated by study sites being open and patients being able to travel to study sites in light of the COVID epidemic. For more information about this study, you can contact us at medinfo@ptcbio.com. We'll also be setting up a study website, www.themightdstudy.com, and we'll be in constant communication with UMDF as well as the patient community about updates on timing when the trial is going to start. And again, we're ready to start as soon as COVID allows us. Thank you very much.
Good afternoon, and thank you to the UMDF for organizing such an amazing meeting. It's heartening to see the number of industry participants in the mitochondrial disease space, something that was unimaginable a decade ago. And I'll be telling you today about the clinical development progress of mitochondrial augmentation therapy for Pearson syndrome. So mitochondrial augmentation therapy is a process by which we enrich patient-derived cells with healthy mitochondria. You can see on the bottom left a confocal image of hematopoietic stem and progenitor cells, which have their own endogenous red mitochondria and have been enriched by green mitochondria using the mitochondrial augmentation process. This is a dose-dependent enrichment, and we've seen this using a variety of methodologies, including enrichment, including looking at protein content by Cox, and using next-generation sequencing with methodologies that can differentiate between exogenous and endogenous mitochondrial DNA single nucleotide polymorphisms. Preclinical studies have shown that mitochondrial augmentation can confer systemic effects. We see that mitochondrial transfer to hematopoietic cells over several months in vivo. Mitochondrial augmentation treatment can raise mitochondrial gene expression in peripheral tissues, such as in the kidney, and mitochondrial augmentation of patient-derived cells enables a higher reconstitution potential of the bone marrow and an immune system changes. So what does mitochondrial augmentation therapy look like in the clinic? Well, for Pearson syndrome patients, this is a non-inherited disease, so the mother can be a donor of mitochondria. We harvest mitochondria from a single blood unit from the mother and cryopreserve these. We later mobilize the hematopoietic stem and progenitor cells to the patient's peripheral blood, which we collect, and then this is augmented using the mitochondria from the mother and reinfused back into the patients. We have an ongoing phase 1-2 study to assess the safety and efficacy of mitochondrially augmented cells in Pearson syndrome patients. These are pediatric patients with a diagnosis of Pearson, which are transfusion independent. And we look at safety and efficacy using the International Pediatric Mitochondrial Disease Score, looking at a change from baseline after a year. The IPMDS was developed by an international consortium of experts over 4 continents and in discussion with patient advocacy groups. And it aims to assess disease burden, how the patient feels or functions, using a range of clinical and functional measures, as well as the patient or caregiver perspective. This is really critical in such a heterogeneous disease such as Pearson or in any mitochondrial disease. The IPMDS was shown to correlate with disease severity and also with other well-known indicators of disease such as the PETI score. And perhaps most importantly, because it was developed with patient advocacy groups, it measures symptoms considered most burdensome by the mitochondrial disease population. So in a Voice of the Patient's report that was summarized and distributed by the UMDF, the symptoms described by patients or caregivers as more bothersome were those such as muscle weakness, chronic fatigue, GI problems. And as you can see, the IPMDS score really asks questions assessing the condition of each one of these symptoms. We also know that Pearson syndrome natural history demonstrates disease progression. And we see this in our patient population in both the currently ongoing clinical trial and in compassionate use patients treated. We know that the average number of organs and clinical manifestations when they were diagnosed were much lower than the number of organs and clinical manifestations at baseline at treatment several years later. And there's about another organ system or clinical manifestation added each year. So the disease burden definitely accumulates, and therefore a stable or improved IPMDS score would be something that would be significant for these patients. This is the current status of recruitment for the clinical trial, as well as compassionate use patients treated. And as you can see, there have been no serious adverse events related to MAT reported. The only SAEs reported were either disease-related, things that were known pre-MAT, or occurred before MAT treatment was given. So we're now looking at how mitochondrial augmentation works and looking at potential biomarkers of clinical efficacy in the peripheral blood. We can see that the copy number of mitochondrial DNA rises in the blood after treatment, and concomitantly mRNA levels rise and ATP levels in the blood. So potentially this can act as a surrogate biomarker to help speed up clinical development for future indications using mitochondrial augmentation therapy. Perhaps most importantly, I have to say thanks to the amazing team that was involved in this work. There were physicians and additional hospital personnel at Sheba, patient advocacy groups, and the families who enable us to try to help treat their patients, and a number of clinicians and academics who are working with us. Really, this is a call for us all to work together as a community to help speed progression of clinical development for mitochondrial disease patients. Thank you.
I would like to thank the organizers for the invitation to present a few insights of the development of Lumevoq gene therapy in Leber hereditary optic neuropathy. Let's firstly look at the disease. LHON is a maternally inherited mitochondrial disease. It is a rare disease with an incidence of 1,500 new patients per year in Europe and in the US. Before onset of the disease, the carriers of the mutations have a very good vision, 20 out of 20. And one day, subjects have a dramatic drop of vision, firstly in one eye, and a few weeks later in the second eye. You see this drop of vision on the left graph. Rapidly during the acute phase of the disease, these subjects reach their lowest visual acuity, which is called the nadir. This is typical of this disease, a rapid and sequential drop of vision, and within 1 year, the patients become blind. The onset of vision loss is on average in young patients between 15 and 35 years old. It mainly affects males. Different mutations on the mitochondrial DNA can lead to LHON. The 11778 mutation on the ND4 mitochondrial gene is the most frequent mutation, representing 75% of LHON in Europe and in the US. It is also the mutation causing the most severe clinical form of LHON. And Lumevoq is specifically targeting this mutation. This is a representation of what LHON subjects face during their daily life. Clinically, this manifests with central vision loss, which is the hallmark of LHON. They get a central scotoma, which rapidly enlarges and becomes very dense, with a dramatic impact on their quality of life. We developed a technology of gene therapy to overcome genetic mutations in the mitochondrial DNA. The gene therapy Lumevoq specifically targets the 11778 mutation on the ND4 mitochondrial gene. Lumevoq is a recombinant adenovirus vector type 2 containing the wild type, the healthy mitochondrial ND4 DNA. Lumevoq is injected via intravitreal injection as it is the best way of administration to reach out the targeted cells, the retinal ganglion cells. Once injected in the eye, the ND4 DNA is transcribed into mRNA in the nucleus. Then the two mitochondrial targeting sequences actively shutter the ND4 mRNA to the mitochondrial surface, where a functional ND4 protein is synthesized and translocated inside the mitochondria to restore energy production. Reverse and rescue studies are pivotal studies. They have a similar design. They are randomized, double-masked, controlled studies aimed at comparing GS10 with a control. Each patient had one eye treated with an intravitreal injection of GS10 or Lumevoq, and the other eye treated with sham. Sham is mimicking an intravitreal injection, but no product is injected. The main difference between the studies is the timing of treatment administration. In reverse, subjects were treated with a vision loss between 6 months and 1 year. In rescue, subjects were treated earlier with a vision loss of less than 6 months. The primary endpoint was the difference of change of visual acuity between treated eyes and sham eyes at week 48. On this graph is represented the mean visual acuity over time for reverse and rescue. In red, the treated eyes, and in blue, the sham eyes. In solid lines, reverse, and in dotted lines, rescue. You see that the treated eyes and sham eyes track together with similar curves of visual acuity over time. When looking at both reverse and rescue studies, you see that the bilateral improvement of visual acuity is coherent between the two studies, with the same shape of visual recovery for reverse and rescue, evolving in parallel, and the recovery starts around the same time point when you consider the time from vision loss. Importantly, patients move from off-chart to on-chart when the treatment starts to be effective. Because we could not reach our primary endpoint in reverse and rescue, and it was the comparison between treated eyes and sham eyes, and the eyes are improving together, it is important to compare these results with the natural history of the disease. That is why we have been running a natural history study called REALITY. And on this graph, you see the reality curve in solid black line, showing that these patients with no treatment have a visual acuity that dramatically decreases over the first year and then progressively decreases with no recovery over 5 years. You see that the shapes of the curves for reverse and rescue have a totally different dynamic, showing a consistent visual recovery over time. LumiVoc was well tolerated. The main ocular adverse event was intraocular inflammation, which was mostly mild, responsive to conventional treatment, mostly with corticosteroid eye drops only, and was resolutive without sequela. There was no systemic safety issues. Since the patients were unilaterally treated with LumiVoc and we got a bilateral improvement in visual acuity, we decided to conduct a non-human primate study to better understand the mechanism of this bilateral effect. The objective of this non-human primate study was to evaluate the potential in vivo migration of LumiVoc active substance from the injected eye to the contralateral non-injected eye. We used a specific qPCR method to detect and quantify LumiVoc DNA in visual systems. We demonstrated that LumiVoc DNA injected in one eye was detected and quantified in the contralateral eye for 5 animals out of 6. We then demonstrated that the transfer of active substance LumiVoc DNA from the injected eye to the contralateral non-injected eye was through the optic nerves and the optic chiasm. Therefore, this demonstration of transfer of active substance from the injected eye to the non-injected contralateral eyes give insights and a potential explanation of a contralateral effect observed in LHON patients in clinical trials. We have compassionate use programs running in Europe and in the US. In Europe, there is a nominative ATU, temporary authorization for use, running in France. 2 patients were already treated, and in the US, 3 patients were already treated through compassionate use. In conclusion, we will be filing for EMA registration in September this year, and we will be moving to BLA for US registration next year. Thank you for your attention.
Hello, everyone. I'm Dr. Peter Stackpool from the University of Florida. I'm a clinical researcher who has dedicated many years of my career studying mitochondrial disorders, and more specifically, pyruvate dehydrogenase complex deficiency, otherwise known as PDCD. I'm the principal investigator of a multi-center, placebo-controlled phase 3 trial, the primary goal of which is to evaluate the safety and efficacy of chronic administration of the investigational drug dichloroacetate, or DCA, in patients with a confirmed genetic mutation in the PDC. Justification for this trial is based on results from a number of open-label studies and earlier clinical trials that suggest that chronic oral DCA is well tolerated by young children and may be particularly effective in improving the clinical course of patients with PDCD. Now, as many of you know, PDCD is a severe debilitating mitochondrial disease affecting approximately 200 to 300 people in the United States. Most of the major signs of PDCD first present in newborns or during infancy and include lactic acidosis, which may be so severe as to be acutely life-threatening, and progressive neurological and neuromuscular degeneration. And imaging studies of the brain may show defects in critical brain structures that are common manifestations of Leigh syndrome, which is a neurological degenerative condition that affects about 25% of PDCD children. Developmental delay and seizures are also frequent complications of PDCD, as are difficulty handling oral secretions, swallowing, sleep disturbances, muscle weakness, problems with balance, and easy fatigability. Feeding difficulties can lead to aspiration of food, resulting in lung infections and necessitating placement of a feeding tube in the stomach or intestine. From the outset, our phase 3 trial of DCA in PDCD has been supported by grants from the FDA and NIH. We've also recently partnered with Therapeutics, a biotechnology company dedicated to finding new treatments for rare diseases. Thus provides us with additional regulatory expertise and financial assistance. The trial is intended for PDCD patients between the ages of 6 months through 17 years at the time they enter the study. Patients are evaluated and treated at 9 study sites across the U.S. Most of these sites are academic institutions well known for expertise in the diagnosis, evaluation, and treatment of primary mitochondrial diseases, such as PDCD. There are no FDA-approved therapies for PDCD. Therefore, to conduct a phase 3 trial consistent with FDA guidelines, the trial must be designed to be double-blind and placebo-controlled, meaning neither the investigative team nor the participating child and family know whether the patient is receiving DCA or placebo at any given time. However, the trial ensures that all patients receive DCA during their participation. The duration of the double-blind study is 9 months. Drug and placebo are shipped directly to the family's home. Participation is free to families. How do we determine if DCA is safe? First, we genotype prospective subjects to determine the most likely dose for them that's both safe and effective. Then we screen them to be sure they meet all inclusion and no exclusion criteria for the trial. Throughout the study, we conduct periodic histories and physical examinations and blood and urine tests as safety checks. We provide these data to each center's institutional review board, to the FDA, and to a group of outside experts in medical ethics and PDCD who continuously evaluate the safety of each participant. How do we determine if DCA is effective? Working with the FDA and with families of PDCD children across the U.S., we developed a unique assessment tool called an Observer Reported Outcome Measure, or OBS-RO. The OBS-RO is a survey that a parent completes electronically in a few minutes at the end of every day. Basically, we ask the parent to assess their child's level of at-home functionality, including neurological function, muscle function, eating, and the like. Each day, after completing the survey, the parent uploads the information to the study's Central Data Coordinating Center. At the end of the trial, it's the findings of the parent-driven OBS-RO that ultimately determine whether their child did better during the time he or she was receiving placebo or DCA. Thus, 100% compliance by patients with completing and electronically uploading a completed OBS-RO survey each day is absolutely essential to the success of the trial. Not surprisingly, the COVID-19 pandemic initially created significant challenges in recruiting, screening, and treating PDCD patients, including travel restrictions and clinic closures. However, we are advancing efforts for remote consenting between families and study sites. We have also arranged for free patient and parent transportation to and from the study sites once opened, and free overnight lodging near each site. Finally, we're exploring the possibility of home visits, if needed, until centers become open and travel restrictions are relaxed. Given the very rare nature of PDCD, we're hoping that this forum will provide an opportunity for networking to make you aware of the current recruitment of patients with this disorder. We welcome patient referrals and will, of course, be happy to answer any questions. It would be great to finally find a proven treatment for PDCD, a truly devastating pediatric disease. Thanks for your attention.
I'm giving an update on a phase 1 clinical trial using citrulline for adults with MELAS syndrome. I'm Fernando Scalia from Baylor College of Medicine. And as background, MELAS is a mitochondrial syndrome that stands for mitochondrial encephalomyopathy, lactic acidosis with stroke-like episodes. And the most common disease-causing variant is this M3243A to G transition in the mitochondrial gene MTTL1 that encodes the transfer RNA leucine. And the prevalence has been estimated to be 16 out of 100,000 in Finland. And we can see here the genetic modification. One of the major features of MELAS are metabolic strokes. And this is thought to occur more in occipital areas in the brain. And they are thought to be associated with mitochondrial proliferation found in smooth muscle and endothelial cells of small blood vessels, seen as these vessels surrounded by the intense blue on succinate dehydrogenase histochemistry. And the thought is that this would be connected with altered metabolism of nitric oxide. And this will lead to changes in the lining of blood vessels, leading to decreased perfusion and stroke-like episodes. And it is known that both arginine and citrulline as amino acids act as nitric oxide precursors with the help of this enzyme, nitric oxide synthase. It has also been observed clinically that arginine supplementation intravenously has led to clinical improvement if given within 3 hours of the onset of symptoms of stroke. And low plasma citrulline levels have been reported in MELAS. This led to stable isotope studies that we conducted in the past and that have been published in adults and in children with MELAS, where we determined that they have impaired nitric oxide production rate and that both arginine and citrulline supplementation increase nitric oxide production, and citrulline supplementation increases nitric oxide production to a higher degree than arginine supplementation. So with that, we obtained funding from NIH through NAMDEC, through the Mitochondrial Consortia, to do a phase 1 clinical trial. And the primary aim here is to establish the maximum tolerated dose of L-citrulline in 24 adult subjects with MELAS at the following doses: 10, 20, 30, or 40 grams per day, divided in 4 times a day. Subjects are enrolled one at a time serially, using a time-to-event continual reassessment method. And the first subject will receive L-citrulline dosage of 10 grams per day for up to 1 month or until a safety outcome is reached. And then the primary safety outcomes and those limiting toxicities are those associated with nitric oxide production. So such as low blood pressure, orthostatic hypotension, hypoglycemia, gastrointestinal side effects, headaches. And the secondary aims of the study are to determine the cerebral blood flow and cerebral blood vessel reactivity using arterial spin labeling MRI. We will also be measuring plasma amino acids at baseline, week 1 and week 4, and we will be measuring plasma lactate at baseline, week 1 and week 4. And in addition, we will measure plasma guanidino compounds by untargeted metabolomic analysis at baseline and week 1 to determine potential arginine toxicity. And the arterial spin labeling MRI will be done at a research facility at Baylor with a breath holding task following visual cues. And this will be performed to measure end-tidal CO2. And an echoplanar functional MRI scan will be used to measure the hemodynamic response to hypercapnia-induced cerebrovascular reactivity. And the maps for cerebrovascular reactivity have been calculated for this trial. And this is a table for the evaluations and the visits to the clinical research visits and the follow-up visits. We'll see that there's a baseline visit, week 1, week 2, week 3, and week 4. So subjects will be on citrulline for 1 month, and then there will be follow-up visits that will take place at week 6 and week 8, which will be the exit visit, with all the labs that actually will be obtained and the collection of diaries and neurological evaluations. We have gone through a regulatory process. We obtained approval from FDA and NIH approval. The protocol has also been approved by Baylor IRB and the Office of Clinical Research at Baylor, as well as the Clinical Research Committee at Baylor St. Luke's Medical Center. The trial is listed in clinicaltrials.gov. We have transitioned to a new data monitoring coordinating center. All electronic case report forms are ready with the new DMCC. We also have training in place for the REDCap system that will be used to collect data. There's a data safety monitoring board in place and an independent medical monitor. And finally, obviously, with COVID-19, we have to pause the study. Now, Baylor St. Luke's Clinical Research Center is reopening for certain clinical trials, but the imaging facility at BCM, at Baylor College of Medicine, is not open for research subjects due to the pandemic. And we expect, that actually the study hopefully will restart in the next few months. Thank you for your attention. The clinical research coordinator is May Ali, and you can certainly contact her at this email address. Thank you very much.